So I use carbon-14 dating, which is an isotope of carbon-12 and that decays in a known rate, and so they measure it and can kind of date back to when the organism was alive. So anyway, so you just need to be familiar with the various forms of dating, but what you don't need to know is I don't have to go into the logistics about how like carbon-14 dating works and stuff like that. So when it says it can be dated, so the age of the rock where a fossil is found. So that's called relative dating, where like sedimentary rock is in layers. And so when you have organisms die, their fleshy parts decay, and you have the bones or whatever stay and again become fossilized sediments. I'll go on top of that, and the sediments then pile on top of each other in layers. They harden, and you get these layers of sedimentary rock. So when you have these layers of sedimentary rock, the oldest layers are deeper in the ground, the newest layers are towards the top. And so when you find layers in these find fossils in these various layers, you can tell the relative age. Like if a fossil is found here versus here, you can tell that this one's newer than this one, died more recent, and so on. And so that's what it means. That's a relative age, whereas this it can be it's more exact, where we're using these isotopes and these radioactive decaying methods and so on that you can actually date the rock and therefore date the fossil. All right, so another thing with fossils is fossils help to bridge the past with the present, and so when it says Darwin's theory explains the succession of forms in the fossil record, succession is the change of forms that we find in the fossil record. And so they've coined this term transitional fossils that are found to link ancient organisms to modern species. And so looking at organisms from the past, looking at them in the present, and then the kind of fossils in between. And so this is a fossil of an extinct whale this is the actual picture of this, the actual picture of it, but seems to have the bone structure of a hind leg limb that is very similar bone structure to organisms on Earth that have a hind leg limb. And so then that leads to wonder, and did they eventually, or were they in the past able to walk on land? All right. And so fossils are found all over the place. Usually fossils are found, like I said, a lot of them are in sedimentary rock, which means that it had to be underwater at some particular point or near water. And so a lot of different people, like paleontologists, can find fossils, but a lot of everyday people as well. And so, oh, thank you. And so this is just a woman named Mary Anning. There's a whole book written about her, and what she lived in England, and on the south coast of England. So this is a picture of this place. Was a fishing village, and on the cliffs were made out of limestone, which fossils are found in limestone, and these limestone cliffs had a lot of fossils in them, and so on. So people will go there to try and find fossils, paleontologists, and so on and so forth. And this woman, who was at the time a girl, she was 12 years old in the early 1800s. She actually discovered, which is pretty remarkable for her age, and so on, the first complete ichthyosaur, which is a reptile. So she had found a whole skeleton of that particular organism, and then also a pliosaur and a pterodactyl as well. And so it shows you that you don't have to be a paleontologist. People discover fossils all over the place, and sometimes you can be looking for them, and sometimes not, and you just stumble upon them. All right, so became a pretty famous 12-year-old. All right, at the time. The second kind of evidence for evolution's kind of category is these things called homologous structures. So homologous structures, what they show is that related species share characteristics resulting from a common ancestry. So these four limbs are examples of homologous structures. The human arm, the cat leg, the whale fin, and the bat wing. Why they're homologous structures, you can see here the color coding of the bones. So the bone structure, not only the type of bones are found in all of the same type of bones, but also the way they're put together, the order of them, and so on are similar and kind of the same between them. What is different though between them? Types of bones are the same, Melissa? The length, okay, so some bones are longer, especially the digits here, and so on, and so the length of them. Why do we suppose that there are those differences? Alan? What was that? To match different needs, okay, so absolutely, and their uses, and so this is another example of structure and function, all right, the structure of it and the function. So while they have the similarity in that they have the same types of bones, the bones and the way that they're put together, those bones are different lengths and so on to meet the needs of what they're used for. And so, so that's what we have here, same structure, different functions. All right, is homologous structures. And then these are anatomical signs of evolution, meaning that these organisms have to be related in some way. They have to share some genetics in there, because what codes for telling your body that we're going to develop an arm with these bones and these orders and so on are genes. And so therefore there has to be some similarities there to direct that development. Third evidence for evolution, this category is called molecular homologies. What this means is you are comparing molecules. What do you think a main molecule that you would compare between organisms to show relatedness would be? Protein. DNA is another huge one. And so remember, DNA codes even for making protein. So in comparing organisms based upon the molecules that they make, organisms can share the same molecules, and then you can see how related those molecules are. So all organisms have DNA. So we have DNA. We also have RNA, and both of those eventually code for making proteins. So if you're comparing between organisms' DNA, what are you comparing? What part of the DNA? Nathan? The nucleotide sequence is specifically the bases of the nucleotides, so the order of those A's, T's, C's, and G's. Same thing with RNA. With protein, what do you think you're comparing? The chain of amino acids. Remember, proteins are chains of amino acids, and so you're looking at that. And so those are your main molecules that we use to compare. And sometimes we look at all of them and looking at organisms to see if they support one another. So if the protein is similar, the sequence of amino acids, is the DNA similar as well, and do they support one another, and so on. All right, so the closer the molecules are, or the more alike the molecules are, that again shows relatedness between the organisms. And so let me explain this particular graph off to the side here. So these bars here and these percentages along to the right here are comparing hemoglobin, which remember, hemoglobin is the protein that's made that is in your red blood cells that's responsible for carrying oxygen from your lungs to your body cells. And so we're comparing the percent of amino acids that are identical to the amino acids in a human hemoglobin polypeptide. So we're going back to comparing hemoglobin in each one of these organisms to a human. So a human to a human. So in looking at hemoglobin compared to hemoglobin, those chains of amino acids, from human to human, it's always the same, pretty much, unless you have some kind of disorder that's going to change that. It's 100% the same. From a human to the rhesus monkey is 95% the same, to the mouse is 87%, and so you can see that, so comparing back to the human. And so what this shows in terms of looking at this particular molecule, it shows relatedness more for humans, related more to these other mammals here than they would be to some of like the reptiles or amphibians and so on. Yes?
Can you also, using like proteins or DNA, distinguish how close things are on like where they evolved from on a genetic tree?
Yes, and we're going to get into that in chapter 25. All right, so just hold your horses there. All right. So yeah, we'll talk about that and how to actually construct a tree and how to use that. Absolutely. Another piece of evidence is that cell structure. So all eukaryotes have similar internal and structure and organization. So what I'm really looking at here is when I say eukaryotes, let's look at the eukaryotic cell in particular. So the eukaryotic cell, say here, have similar internal structures in organization. So what does that mean? It means that all eukaryotic cells have a nucleus, they have ribosomes, they have DNA in their nucleus, they have mitochondria, rough ER, smooth ER, Golgi apparatus, and so on. Relatedness between different types of cells, though, this is a quick little review of endosymbiosis, or this endosymbiotic theory. Proposes that eukaryotes, remember, originated when larger prokaryotes engulfed them, and they became part of the smaller prokaryotes. That's what the simulated means. What two organelles were involved in this? What two organelles? Give me one, Roman. Chloroplast, and another one, Nathan? The mitochondria. So both of these are hypothesized to have been prokaryotic organisms.
Yeah.
So both of these are hypothesized to have been once free-living prokaryotic cells. And so then they were engulfed through cytosis into the cell. So let's just review here evidence of this. All eukaryotes have mitochondria for energy transformation. So that's one thing that we have in common between all eukaryotic organisms. So there's some relatedness between every single eukaryotic organism on Earth. The mitochondrial DNA originates solely from the mother. So the mitochondria has DNA, and remember it comes from the mom, because remember the sperm doesn't donate the mitochondria when the zygote is formed. All right. Autotrophs, organisms that make their own food, have chloroplasts for photosynthesis. And I'm going to have here, and the chloroplasts have DNA as well. So the chloroplasts were probably also once free-living organisms. And then both the mitochondria and chloroplasts grow and divide on their own, kind of like bacteria do. And they both resemble modern bacteria, meaning that bacteria basically have DNA and they have ribosomes to make proteins. That's basically what's inside of them. And that is the same for mitochondria and chloroplasts have ribosomes and DNA. All right, so every single eukaryotic cell has some kind of relatedness, anything that's made from eukaryotic cells, because they're made from these same things. One proof or piece of evidence for the endosymbiotic theory is the discovery of this red seaweed called Porphyridium. And so what this red seaweed does, it says it has a plastid. I want to talk a minute about what plastids are. Notice that the word chloroplast has that plast in there. So plastids are a group of organelles. Chloroplast is an example of one of those. And so there are other types of plastids. Chloroplast is just an example. So this porphyridium is red. It has these plastids in it like chloroplasts. There are other types. There's chromoplasts and all different types of plastids. I'll just stick with chloroplasts here. And what they found is that when they studied this red seaweed, so if this is a seaweed cell, so that's the cell from the seaweed, and it has a nucleus, so this is the nucleus, and inside the nucleus is the seaweed's DNA. But the seaweed cell also has these plastids, so I'll just draw kind of here like a chloroplast here. And remember, what do chloroplasts have? They have their own DNA, right? So here's the chloroplast DNA. Chloro DNA. So they have their own DNA. So this is what we're talking about here. What they did is they found the seaweed and they took these cells and took the seaweed DNA and compared it to the DNA in the chloroplast. And then they took it one step further and they took bacteria. So the bacteria has nothing to do with the seaweed. So this is a prokaryotic cell. So here's your bacteria. And bacteria, remember, have plasmids and then they have their big circular chromosome. So here's the DNA and they have the plasmid. That's what bacteria have. So what they did is took this chloroplast DNA and compared it with this and the bacteria. And what they found is that even though this chloroplast is part of the seaweed cell and part of the seaweed, when they compared the DNA, this DNA inside the chloroplast was more like the bacteria's DNA than it is to the seaweed itself that it's part of. So that, can somebody tell me how that provides evidence for this endosymbiosis theory?
Show that like the DNA in the plasmids of the seaweed are actually made by the seaweed because they don't share the same DNA. It would be some like other organism origin.
Okay, yeah, all right, and so this gives evidence that this chloroplast was once a free-living organism, prokaryotic cell like bacteria. All right, and so that also shows, some relatedness between prokaryotic and eukaryotic cells as well. And so this is just a picture. We've already talked about it, so I'm not going to spend much time on this. The endosymbiotic theory. All right, next evidence for evolution is looking at embryos. And the embryos, this is just one piece of evidence out of the many. So here we have comparing human and chick embryos. Clearly, as adults, there are a lot of differences between humans and chickens. All right. But what this is showing here is that there must be some genetic similarity at some point between chickens and humans embryos because they have some of the same features in the embryonic form. They are not shared in the adult form, okay? So looking at different stages of development. And so at some point in human and chick development, there's a post-anal tail, and we have these pharyngeal pouches that in fish actually develop into gills. They go away as we get as we develop here, and these pouches actually develop into certain parts of the it's called the endoderm of the the inside of your neck and so up through the tonsils in the middle ear and so on and so forth. And so they develop differently, but they're both present in different organisms and have ultimate end. The end result is different. And we do not have a tail eventually as we continue to development, but we do at some particular point. So looking at embryos and looking at similarities at different stages of life and so on. The next one is a vestigial structure. So vestigial structures, as it says, have no apparent function in living organisms, but the living organism has these structures. So the thought is that they resemble structures held by ancestors that are extinct, that those structures used to be used or useful. So we see these in the fossil records. So for instance, there have been fossilized skeletons of snakes that have bones that resemble the pelvis and leg bones. We've seen in real life pythons. This is a python that has a vestigial appendage. It's really hard to see here. It's a little thing that protrudes out of the backside here, and it's a hind limb bud. Early on in development, before even our arms and legs develop, we have little limb buds that eventually grow into our arms and our legs. So this would be a vestigial structure, but clearly they have some type of genetic genes to grow that.
Would an example of a vestigial structure in humans be like our appendix or their molars?
Yes, so a lot of people use the appendix as an example. The appendix does have, we can live without the appendix. It does have there's somebody at the door. They found that there's some,
I need to freeze for my collection pictures.
Okay, Liam. I know you just asked a question. I was just answering this question. Now he's got to leave. You made your
holiday, yeah, sure.
So, but yes, that's right, okay? And so, so a couple of other examples, or another example I was going to use was the blind mole rat. These guys live kind of near the Mediterranean. There's some in Israel. The blind mole rat have eyes, but they're small eyes. And get this, the eyes in their head are actually, it's grown over, skin grows over it, and so it has skin and hair, fur over it. But there's eyeballs underneath it. All right.
Can they actually see? They're just seeing it.
No, they can't see either. Yeah, so their eyeballs are there. They have the genes to make the eyeballs. They're not useful. They can't see through them, so it's not like they're just seeing black. All right, yeah, that's a good question. And so that's considered a vestigial structure, and thought that at some point in the past those eyeballs were useful. All right, and so those are vestigial structures. All right, and so, and then the last piece of evidence is called biogeography. And so if we dissect this word geography and bio means living, so we're talking about living things and where they're at and found on the earth, the geography of them. So the geographic distribution of a species. So what we found is that in studying species that live closer together and in closer proximity are usually more related than species that are far apart. And it doesn't even have to be individuals of the same species, but within the same species, that's true. So there's more relatedness between individuals of the same species in certain areas. That's why, like, I'm going to use Australia as an example here. So Australia has what are called marsupial mammals. Hold on, pause this for a second. Let me get zero. I realize
I can only do
so I was getting ready to talk about marsupial mammals. Kangaroos and koala bears are an example of that. Does anybody know what a marsupial mammal is? What does that mean? It has something to do with pouches.
They have.
Nathan? Yeah, shh.
They like, I don't know if it's like an egg or like an early birth, but like the baby has like, it comes out like really, really small and then like lives in like a pouch on the outside.
And actually finishes its development in the pouch, all right? So it's not, the pouch isn't just a cool way to carry around babies, all right? Which is what some people I think think. It actually it finishes its development in that pouch, the baby does. And so that's different than us. We are what we call placental mammals, where the baby finishes development inside the mother, and we have a structure called a placenta that helps to feed and nourish and bring oxygen to the baby, and it finishes the development within, and then the baby is born developed, all right, and so on. And so what that's saying, though, is that in Australia, you have a huge diversity of marsupial mammals in Australia, on this continent of Australia, but not really around the rest of the world, all right? So therefore, there's relatedness, they're close together, relatedness between them and other organisms on Earth. And so species that live closer to one another tend to be more closely related than those that do not live closely to one another. And so Australia is an example of that diverse population of native marsupial mammals, but almost no placental mammals. And that's opposite for us. All right, and North America. Okay? And that is chapter 22. Before we talk about chapter 23, I want to talk about the article that you picked up yesterday that, remember, I said you could hold on to and wait till today to do. Maggie, did you get my text?
Yeah, I did, but I still did.
I was like, no, I better send this out because I didn't tell Maggie. All right. So let me explain what you're going to do here. If you need it, it's up here in the Monday. If you look at the backside, it has some things written on here. I'm going to explain what you need to do. The title is Evolution Hidden in Plain Sight. So what this is going to do is talk about evolution in that we can see it happening. And it's going to talk a little bit about the things that we've talked about, like it's going to talk about mutations, and there's some gene duplication in there, and so on. What you're going to do is on the back. See where it says closed reading strategy. So number 1 says go back through the article and using a method that's most attractive to you, different colors, writing in the margins, underlining. You're going to do some annotation. And it says indicate where in the article each of the following can be found, and so therefore there's those things that you're going to focus on, and you can write and annotate on it. All right, so that's what I'd like you to do. For number 2, I'm going to change number 2. So number 2 asks you to list any questions you have. That's not what I'm going to have you do. What I'd like you to do for number 2 there is just write a 25-word or less summary of the article. So I want you just to give the brief idea of the article. What was the article about? In a nice, concise manner. And then underneath consideration topics, I'm only going to have you do 1 and 2. You can do them all. Okay? So this is 4 tomorrow. Okay? All right, so let's look at chapter 23. So 22 was an intro to evolution. So what we're going to look at next is how populations can evolve. So yesterday we defined a population as a group of species of the group of individual species of all of the same species living in a particular area. And so that's the smallest unit that can evolve. So like an individual can evolve. You're born with the genes that you have. So individuals don't evolve unless, of course, you're a prokaryotic organism, because prokaryotic organisms can get a plasmid and then change the genes, you know what I mean? So then they change the organism. But multicellular organisms, that's not the case. So the smallest unit that can evolve is, we say, a population, because the population is individuals of the same species. It's the next kind of hierarchy in the ecosystem is a population. So study of how the gene pool of populations change over time is what population genetics is. So what we do is, in studying this, is when we remember the gene pool is all the alleles in all members of a population. So I put this up on the board yesterday. If we have these 5 individuals, this would be the gene pool, and each one of these individuals have 2 genes, right, for a particular trait. So all of these genes comprise of the gene pool. So when we have an organism like ourselves that's diploid, in a diploid species, each individual has 2 alleles for a particular gene, and the individual can either be homozygous or heterozygous. So it can be homozygous dominant, big R, recessive, little r, or heterozygous. Whose genotype do we know for sure always? By looking at them? Homozygous recessive, right? the dominant individuals that show the dominant trait, we don't know if they're big R, or big R, little r by looking at them. All right, so that'll become important later. All right, and then remember, yesterday we talked about the fitness of an organism. So the fitness of an organism refers to the contribution it makes to the gene pool of the next generation relative to the contributions of other members of the population. So what does that mean? If an organism has a good fitness level, so it's fit for their environment. Let me shut this door. Remember, what does that mean? They have traits that are good to help them to survive in that particular environment. So what this paragraph means, this sentence means is, if I'm fit for my environment, I have genes that help me to survive. That means that I'm going to live, I'm going to have kids, and I'm going to pass those good survival genes on to my kids, and I'm going to change, and my genes are going to be part of the next generation, the next gene pool. If I am not fit for my environment, then most likely I may not reproduce. That means I'm not passing on my genes to the next generation, so I am not contributing to the next generation, and my genes aren't going to be part of that gene pool. So that's what it means by looking at the contribution relative to other members of the population. So in other words, a more fit organism survives and has viable offspring. Viable means they thrive, they're living offspring. So it's not going to be helpful if you have offspring that, are weak in some manner, are going to die. It doesn't matter if you really reproduced in that particular case. And then remember, the basis of natural selection is that there is genetic variation. And it happens, there's genetic variation in all populations, not just some. Some can have more genetic variation than others, but there is genetic variation in all populations, which is the basis of natural selection. So we're going to study the evolution of populations. And how we do that is we use something called the Hardy-Weinberg theorem. This is these two scientists, Hardy and Weinberg, came up with this. And what we're going to do is talk about how a population changes over time. The genes change from one generation to the next. That's what is the definition of the population evolving. So what we're going to do is look at kind of like a controlled experiment. We're going to look at a population that's not evolving and compare it to one that is. So the Hardy-Weinberg theorem is used to describe a population that's not evolving, so then we can compare it to populations that are. So we're going to spend a few minutes and talk about what it means for a population to be not evolving. So what that means, the frequencies of alleles and genes in a population's gene pool will remain constant over the course of generations unless they are acted upon by forces other than Mendelian segregation or recombination of alleles. So what does this mean? So normal reproduction, like when we make egg or sperm cells, we have, your genes, your chromosomes come up, they can line up in two different ways, they segregate into different egg and sperm cells, and then the egg cell is fertilized by this particular sperm cell. And you do get some recombination just due to sexual reproduction. But this is talking about other things that are affecting the gene pool. So when it says the frequency of alleles and genes in a population's gene pool, let me talk about what that means. So let's look at this particular population here. There's only 5 individuals, so it's a very small population. These 5 individuals, each of them, for whatever trait we're looking at here, have 2 genes, right? So the gene pool of this population has a total of how many alleles or genes for this trait? There's 5 people. 10, right? 2 for every person, okay? So there's 10 total alleles. So if we want to figure out, it says the frequencies of alleles, we have 2 alleles here. We have the big R allele and the little r. Oh, it's Leo. At least I hope it's Leo. All right, frequency of the big R and the frequency of the little r. What we do is, so in this population, our total alleles are 20. So to figure out the frequency of, let's say, the big R allele, out of that 20, we figure out how many are big R. So in this particular case, there's how many that are big R? 4, right? 1, 2, 3, 4. All right, so 4 out of that 20 are, 10. Did I confuse you?
Yeah.
Sorry. My next example is going to be 20, so that's why I got ahead of myself. All right, so 4 out of the 10, sorry. And then that equals to, if I divide that out, what decimal? 0.4. And frequencies are written in decimal format, kind of like probabilities were. So what this really means is, what this is telling you is out of all of the alleles, really 40% of the alleles are big R, okay? And so therefore, 40% are big R. That means that the rest of them are little r, so we would say that 60% should be, and we can figure that out mathematically because there's still 10 total alleles. And when we look at that, there are 6 little r's, 2, 4, and 6, and that is 0.6. These guys should always equal 1 or 100% because in this case here, all the alleles for this trait have to be a big R or a little r. And those are what we call allele frequencies. So what we say is when a population that is not evolving, what that means is that over generations, so this population reproduces, and when we look at the frequencies of the genes in the population, it should still stay 0.4 and 0.6. And the next generation and so on and so forth. So they're always 40% of the genes are big R, 60% of the genes are little r. It does not mean that the genotypes have to stay the same. Can I have different genotypes in my next population but still have 40 and 60? Absolutely. All right, so for instance, can somebody tell me how I would do that? Give me an example of how I would make it different population but the frequency is the same.
First two both be homozygous dominant and the rest be recessive.
Like this? Absolutely. And this would still give me 60% a little r, 40% big R, but my genotypes aren't the same. So we're not talking about the genotype staying the same, we're talking about the frequency of the alleles. So if they stay the same, we would say that this population is not evolving. So the situation in which the allelic frequencies within a population are not changing is referred to as what we call the Hardy-Weinberg equilibrium. The Hardy-Weinberg equilibrium means then that the big R and little r is always staying the same. And we would say then this population, we'd say, oh, this population is in Hardy-Weinberg equilibrium, all right? So let's talk about what needs to be true for this to happen in a population. So there are 5 things that need to be true for a population for their big R and little r frequencies not to change. So the first one is the population must be extremely large so that no genetic drift can occur. I'll talk a little bit in a little bit here about genetic drift here, but let's just talk about the population must be extremely large. So let's talk about the opposite. Can somebody tell me how a small population would allow the population to evolve more than a large population? What could happen in a small population that would allow it to evolve more than a large population?
One mutation is changing a large percentage of the population. So if you have 10 or something, one of the changes is 10, where if you have 100 or something, one changes.
Yeah, absolutely. So one individual affected like by a mutation is a larger percentage of a population of 5 than it would be a population of 500.
There's also like in a small population, the ability for like one individual to mate with like pretty much a lot of more individuals than like say another one. So like for instance, I don't know when it was, like whenever Genghis Khan was a person. Like now, like 10 or 5 or 10% of like all Asians now are like directly related to him. And that's like
Yes.
he got around.
All right, thank you for sharing. All right, absolutely. So using my example up here, if, let's say something happened to like these two individuals here, let's say they got a disease or they got killed in a tornado or something like that, all right, and I have drastically changed the frequencies of my alleles here just by changing two people or two individuals affected. Whereas if this population was 1,000 individuals and the same thing happened to those individuals, it wouldn't change it that much. So large populations, big deal in keeping Hardy-Weinberg equilibrium. There must also be no migration or gene flow in the population. So migration, there's two different kinds. There's immigration and emigration. So that means that people coming in. Immigration is people coming into the population. So if this is my population and 5 little r people came into the population, have I changed my frequency? Absolutely. We've increased the little r frequency and decreased the big R frequency. Same thing if emigration, where you leave. So if all the big R individuals left, we changed the big R frequency and the little r frequency as well. So no migration. So the population has to stay, whoever's in it, that's just staying in it. There can be no mutations. So So looking at my hypothetical population here, if it's a small population and this big R, let's say, mutates to a little r, then I've changed my frequency of my big R and little r genes. So we have no mutations. Random mating must occur. This means that there must be no criteria that individuals are looking for in choosing their mate. So therefore, they will mate with anybody, all right? And so that's what random mating means. So can somebody tell me how the opposite of that, having non-random mating, could lead to gene change? How could non-random mating lead to the frequencies changing over time? Nathan?
So if you have a population with two distinct traits and one of the distinct traits is like desirable and one isn't, and the one that doesn't have the desirable trait or has the desirable trait slowly disappears. So the one like
That's right. That's right. So if it's non-random, is that if there's a desirable trait that the individuals are looking for, and they mate with individuals with that desirable trait, then that desirable trait is going to go up in the population over time, whereas traits that are not desirable will go down because individuals aren't going to mate with them. And so that's going to change the gene frequency. And so that's so there must be random mating, so therefore all characteristics are, everybody's on an equal playing field, all right, in terms of that. And then lastly, no natural selection can be taking place. So by the same token that we just talked about non-random mating, remember natural selection, the environment, that if you have two different traits here, and one trait helps you survive more than another trait, this trait that's helpful for survival is going to be passed on. Those individuals will reproduce, pass those traits on to the next generation, and that trait goes up in frequency, other traits go down in frequency. So if you have no natural selection, what that's saying is all traits have equal survival rates, all right? So there's no trait that's better than another trait to allow the individuals to survive in their environment.
Wouldn't the human population then be like the best example of a Hardy-Weinberg equilibrium? Because it meets like all 5 criteria the best.
Yeah, I wouldn't say no mutations though.
That's true, but it usually is just within like one.
And I don't know about the random mating. I think that there's criteria.
Yeah.
I don't know.
And there's probably exceptions to that, but as the human race as a whole, I think.
Did you really say that about like the entire population of like
I
the
didn't
globe?
say the entire, but I think the majority probably, that they're looking for certain traits in a mate. Yeah, I do believe that. But I'm not going to argue about that. All right, so we're not going to have that discussion. All right. So, how likely, this bringing me right to my point, how likely is that most populations in the wild that they meet all of these requirements all of the time simultaneously? Not very likely. And so therefore, this Hardy-Weinberg equilibrium, populations in Hardy-Weinberg equilibrium, this is more of a theoretical population, that if there's no evolution occurring, no change over time in the population, that these 5 things would need to be true. But generally, in the wild, there is some of those going on. All right, and so that's a hypothetical, and it gives us a point of comparison. So what we're going to look at next is some of the things that affect the Hardy-Weinberg equilibrium. And so I had wrote over here, for number 1, the population must be extremely large so that no genetic drift can occur. And so, so let's talk about what genetic drift is. Unpredictable chance fluctuations in the allelic frequencies of a small population. Small is a key word here. Small population from one generation to the next. So I'm going to go through 2 examples of this. So the first one is called the bottleneck effect. So this is a type of genetic drift. A sudden change in the environment, like an earthquake, a fire, I used the tornado example earlier, drastically reduces the population. And it's totally random. So you have, like, you know, think about an earthquake, it doesn't pick and choose who's going to kill, all right? So therefore there is a randomness. And so certain individuals survive, but they're totally random which do. The survivors then may have a gene pool that no longer reflects the original population's gene pool. And so that would be like in my 5 people up here, if a tornado came and wiped out these guys here, the remaining individuals do not reflect the genetic diversity that the original population had. There's way fewer big R's than there are little r's, okay? So that's what that means. That's called the bottleneck effect. The second way that genetic drift can happen, where you get a change due to small population size, is called the founder effect. A few individuals become isolated and establish a new population. It's called founder because they can found a new population by moving away from the old population. And so, so the gene pool is not reflective of either the source or population, or you can write original population, and so it changes. So I'm going to give you an example here with these flowers. Notice here these flowers on the left. So we're going to look at this this is the original population. We're going to look at it through 3 generations, showing genetic drift. So here's your population. There's 10 flowers here. And it tells you down here that P, we're going to learn that what P is in just well, probably tomorrow. But it's the frequency of the red gene allele, and Q is the frequency of the white gene allele. These flowers exhibit what kind of inheritance? Look at this is incomplete dominance. To be red, you have to have 2 red genes. To be pink, you have to have red and white. To be white, 2 white genes. So therefore, they use this as an example because there's never a question then about the individual's genotype. All 3 phenotypes have their same genotype. Hold on, guys. What I'm going to have you do, they wrote that the frequency of the red and the white gene are 0.7 and 0.3. What I'd like you to do to the left of this is show the how they figured that out. So I want you, like if I were not to give you 0.7 and 0.3, I want you to figure out and write next to it how they got that. This is where I got my denominator. I was thinking of this example. So you have to look up here. So, you know, that was the transfer from PowerPoint. Yeah, I didn't know that it didn't transfer the genes onto your picture. So you have to, like the C's don't have the sign.
Oh, that makes more sense.
Yeah, I didn't realize, you guys, that the second C on your, underneath your flowers don't show what the gene is, so you might have to look up here, or, I don't know if you can tell the colors, like I just wrote my red, pink and white.
Okay.
Alright, and then after you do that, then this is where we'll stop. What is she? She's like the queen of the I like it too, I use them with great, but mine are starting to.
You don't want that?
I'll see you guys tomorrow.